Good morning, everyone from Baltimore. This is Mohammed Mansoor Mohyuddin. I'm the current president of IXA, and on behalf of the Transplantation Society and International Xenotransplantation Society, I would like to welcome you to this webinar, which is jointly hosted by these two societies and associations. Here we are gathered here today with a panel of experts to discuss some of the papers that were jointly published in the Transplantation Journal and in Xenotransplantation Journal. We have Dr. Jonathan Bromberg, executive editor of Transplantation here, and Leo Buehler, who's the editor of Xenotransplantation here. So IXA, the International Xenotransplantation Council, have been compiling data for many years, and finally they are able to present this data to you in form of multiple papers which have just been published, and we are gathered here to discuss those papers by the lead authors. This project was led by Dr. Hawthorne, who gathered all these authors to get us started. So we have invited these authors to briefly discuss these papers and the highlights of these papers with you. Every author will have about 5 minutes to give some highlights and 3 minutes each for some question and answers. And then after all of them are done, we'll have a panel discussion, and you are welcome to ask questions through the chat to the question answer session after that. And then we'll wrap up. I would like to invite my co-chair, Dr. Jonathan Bromberg, to say a few words, and then we'll start.
Thank you very much, Mohammed. It's a pleasure to be here. This is really an all-star cast of speakers and an all-star series of papers that have just come out in transplantation that have all come from IXA. Just to briefly mention them, one paper is on the history and current status and regulation of Xeno. The senior author is Dr. Hawthorne. Next paper is on current data and future directions. The senior author of Hao-Wei Hu. The third paper is on kidney xenotransplantation, and the senior author is Dr. Meyer. And the last paper is on infectious disease considerations in Xeno, the senior author, Dr. Fishman. And there is an overall commentary by Joe Tector to give a roadmap of all of these papers and what they represent. So I won't take any more time, and I'm very anxious to hear all of the speakers. Thank you very much.
So let's get started with our first presenter, Dr. Wayne Hawthorne. He's the immediate past president of IXA. He's a professor of surgery, and he's an authority in the field of islet, allo, and xenotransplantation, and has led all this effort to gather all this information. So without further ado, Wayne.
Thanks very much, Mohammed. I'd like to thank the organisers, particularly Jen Lee, for getting this together for the Education Committee of TTS. Underpinning all this, as we've just heard from Mohammed, is an overview that the IXA, in combination with TTS and the WHO, have been working for several decades to develop guidelines, guidances, and white papers to ensure that we underscore and undertake clinical xenotransplantation in an efficacious, regulated, safe, and effective way. Now, The first papers come about basically because we felt that we had to give some sort of overview. There's been a number of papers over the years giving history of xenotransplantation, but people seem to forget the long-term history and our forebears who did huge amounts of work to lead us to where we are now. As you'll see, there's a huge number of authors in these publications, and they are mostly people who are on the IXA executive or council and have decades of research experience. Now, underpinning this, as I was saying, we go back not decades, but even up to several hundred years ago. And I'd like to bring up one of the very first published works was from John Baptiste Denis. And he was famous or infamous because he transfused patients with blood. And he was the first doctor to kill a number of patients with xenotransplant transfusions. Which meant that the French government implemented the very first moratorium against xenotransplantation. And as we saw in the early 1990s, going back then into the 2000s, we had a number of moratoriums placed on xenotransplants from the clinic around the world. Some of our forebears and people that bear the name of a number of awards. Keith Reemsma was very famous because of the first xeno kidney transplants. These were performed from rhesus monkeys, baboons. The same with our very first and probably the most famous baboon heart transplant into baby Faye by Leonard Bailey. Keith Reemsma has an IXA award named after him. The other person that we have an IXA award named after is one of our godfathers more recently, Professor David Cooper, who came up with the discovery of the alpha-gal epitope. And alpha-gal, as you all probably know, is the thing that was the major barrier to prevent us from transplanting across from porcine into humans because it caused hyperacute rejection. So we've jumped from non-human primates to porcine xenotransplants, and we'll get a little more background in a minute on that. Obviously, we've moved forward to where we use the alpha-gal knockout. And I'd like to mention two great Australians, a very famous Professor Ian Mackenzie and Tony Dapachet, who were the first in Australia to knock out alpha-gal and allow us to do our own xenotransplant research in non-human primates. And of course, we'll hear a lot about the first heart and kidney transplants coming up. Underpinning all this, though, the international community were concerned that we had to have absolute guidance and authorization by the regulators to ensure that we had appropriate regulations, but informed guidance from the international experts to ensure that we wouldn't have any issues in relation to, and certainly the big one that WHO and internationally people were concerned about was that of porcine endogenous retroviruses. So much so that we had a number of moratorium lodged to prevent clinical xenotransplant moving forward, and we can talk about that later, or there may be questions. Underpinning this, then the International Xeno Association set up, with conjunction with TTS and WHO, a number of communiques. Starting back in the early 2000s, we had a focus from the WHO, the Director General, requesting that we set up a registry of xenotransplant clinical work. and a number of communiques developed out of that. Certainly in 2008 was the first WHO Changsha communique, second in 2011, and we held our last one in 2018. And we're looking at moving forward to doing another one at the Congress in Geneva in September this year. Underpinning this again is ensuring that we get information from various community groups, as well as the public, as well as those patients that require transplantation. So moving forward to that, part of the paper that we have for the history and regulations, we actually are looking at, as in the xenotransplant community, how we can address this with public consultation. And the table that's presented in this paper is actually focusing on how we're going to move forward with our public consultation work from the IXA. Not just to mention that, there's been a huge amount of work developed around the pigs themselves, developing the correct types of genotypes to actually be able to transplant. And we'll hear a little more about those moving forward as well. But I guess the big thing that has jumped us miles ahead and now allows us to do our clinical work is that of CRISPR-Cas9 and multiple transgenes. But also, what are those transgenes are very important, as is the non-human primate and research-based work that we do. So in summary, IXA Council, along with TTS, WHO, and the regulators such as FDA, TGA, EMU, have all been working for decades to try and ensure that we can safely move forward to doing clinical xenotransplantation. And in the next presentations, we'll hear something about that work as well. I'd like to ask anyone viewing in to become a member of IXA if you have a keen interest in xenotransplantation. And I'll leave it there. So thank you.
Thank you, Wayne. And just to remind the audience that please put your questions in the Q&A box, not in the chat box. So Wayne, you mentioned about the moratorium on xenotransplantation. Is this moratorium still existing, and which countries is
No. So I guess, as I said, the whole thing was that the WHO wanted to ensure that there was no pandemics. So at the time, the regulators like the FDA, the EMU, the TGA, which is Australia's, set about enforcing regulations, and then decided, because of the absolute risk, potentially the theoretical risk to patients and the community, the US government in the 1997, 1998 had a moratorium on performing clinical xenotransplantation. However, they dropped that in 1999, but at the same time, they banned all non-human primate and primate organs to be used in xenotransplantation. Following that, the EMU, the European Council, in 2003 had a moratorium all the way through for several years until they came up with accredited regulations. And following again, our own in Australia, a little behind the times in 2005 to 2009, we were not allowed to do clinical xenotransplantation. And this actually affected our research funding, I guess, which is the big thing. When you stop practice moving forward to the clinic and stop non-human primate research, you actively will prevent the potential for us to move forward to the clinic.
Thanks, Wayne. And we have more questions, but we'll keep it for the end. And I would introduce now our next speaker, Dr. Raphael Meyer, who's at my institution at University of Maryland. He's an abdominal transplant surgeon. He's also a counselor and our young prospect to lead in in the future in the field of xenotransplantation. And he will talk about the the position paper on the kidney. Go ahead, Raphael.
Good morning, good afternoon, good evening, everyone. Thank you so much for the introduction, Dr. Mohideen. I have the privilege to present the position paper on kidney xenotransplantation by the IXA. And I had the privilege to work with this fantastic group of people that you can see on the bottom of the screen, including all council members of the IXA. On this position paper. On the left side of the screen, this figure displays the burden of end-stage renal disease. And as you can notice, without the option of transplant, which is reflected by the survival curve in red and black, the survival of our patients is very poor, and added value of living donor and deceased donor transplant are fantastic for the patient, improving survival from 50% at 5 years to all the way up to 90% plus. This is the rationale of exploring xenotransplantation for our potential kidney transplant patients. On the right side of the chart, you can see the evolution over the years of the result and the survival of pig kidneys transplanted in non-human primates in a life-sustaining fashion. And we witnessed with the development of the multiple genetic modification and immunosuppressive regimen, an increase in the survival of those pigs up to 1 year quite consistently and up to 2 years in some cases. So in this figure, I present what happened over the last few years, starting in 2021, and which was mostly initiated by very few, like Dr. Mohideen at our center with a heart xenotransplant and others with some decedent transplant of genetically modified pig organs. And on the left side of the screen, we see a couple of attempts with decedents, so brain dead donors, to test for short-term viability. And these experiment was quite successful and gave us confidence to progress towards transplanting a living patient, and that you can see on the right side of the screen, where MGH and NYU transplanted a few patients each, and quite successfully, with one of the patients still having a functional graft over 120 days post xenotransplant and MGH. Here is the summary of recommendation from the IXA group on kidney xenotransplant regarding immunosuppression, the source pig, recipients, and post-transplant monitoring. Immunosuppression mainly, of course, that was used in most of the cases involving living individuals with T cell depletion and B cell depletion. Very important to add some complement inhibition, and those were of course added with the quite standard immunosuppression regimen that most of our patients receive with the MMF, calcineurin inhibitors, and steroids. The question mark remains with how much we need to turn down inflammation with anti-TNF, IL-6, IL-8, and others. Another interesting question I find is the use of ex vivo perfusion in the intention to reduce initial inflammation after transplant. Source pig, most of the experts agree on the fact to use triple knockouts pigs with various transgenes, meaning that we add some human genes in the genome to regulate the complement, to regulate the coagulation, and to attempt to reduce inflammation. The selection of recipient is really one of the key part, and we've learned so much over the last few years on that perspective. And basically, it seems like two categories were used recently. Some hyper sensitized patients with no option for allotransplant because they have a very, very high PRA of 100%. And other patients with biologic age, which is between 60 and 75 years old, and we will have to wait a lot of years before getting a transplant. So there are some options. It looks like the hyper-sensitized patient is maybe not ideal because there are some cross-reactivities with anti-pig antibodies. And lastly, post-transplant monitoring. Of course, it looks like there is a need for developing new platform, including, for example, precision diagnostics and very, very precise assessment of biopsy and other biologic markers to follow our patients, to give the maximum precision and details in the analysis of how their graft do, because within a few days they can develop quite severe rejection. So, and I would emphasize that so far almost actually all the patients had rejection episodes, and this is something we have to define as a community. Thank you so much for the opportunity, and I will be happy to answer questions.
Thank you, Raphael. One question is that we have all heard about upcoming clinical trials. Would you like to comment on, what are these trials and what to expect from them?
Yes, so there are two trials regarding two different companies. One is United Therapeutics, the other one is eGenesis, and they've been granted, I think we have 6 patients potential with the trial with the company United Therapeutics, and 3 compassionate use with eGenesis companies. There is also, I think, as a side note to mention that there is a trial with one of these companies with regards to the liver ex vivo perfusion.
Okay, great, thank you. And we'll hold the other questions till the end. And I would like to introduce our next speaker, Professor Jay Fishman. He's the past president of American Society of Transplantation and president-elect of International Xenotransplant Association. He's the professor of medicine at Harvard Medical School and director of the Transplant Infectious Disease Program at the Mass General Hospital. He's associate director of MGS Transplant Center, and he's a leading expert in infectious disease, which is a major concern among all the regulatory agencies. So, Jay, please take over.
Thank you, Mohammed and Jonathan. It's a pleasure to join this discussion and to talk a little bit about the paper, the IXA position paper on infectious disease considerations in xenotransplantation, which I did with my long-term friends and colleagues, Joachim Denner and Linda Scobie. The notion of xenotransplantation, of taking a pig organ and putting it into a human to treat organ failure, raises, for those of us in infectious disease, concerns about the potential transmission of pig organisms, potential human pathogens or zoonoses in the recipients, particularly because those recipients are immunosuppressed. And so many years ago, in the 1990s, my lab began studying a number of porcine organisms, and we cloned the porcine endogenous retrovirus, which unfortunately led to the moratoriums that Wayne Hawthorne described a little bit earlier. It has turned out subsequently that this particular virus, the porcine endogenous retrovirus, is less infectious, although there are receptors in human cells, appears to be less infectious than originally feared, and in fact can be excluded by genetic techniques. But let me talk about the key concepts from the paper, which is that all animals carry microorganisms, and the risk of infection in a recipient is determined by the risk of those microorganisms from the donor and in the recipient and from the environment, but also by the immunosuppressive regimen that we use to prevent graft rejection that Raphael described just a moments ago. And because we're using more intensive immunosuppression, the concern is that perhaps we'd see more infections in xenotransplantation than we have in allotransplantation. In fact, as we'll talk about, because of the screening and the time we have available to screen potential pig donors, there may be a reduced risk of infection transmission from the donor to the recipient because of our care that's taken with these xenografts. But in fact, to do the testing on the breeding herds and on the recipients, we need new, validated, specific, and sensitive detection methods and testing strategies to deploy to make sure that there are no organisms in the swine that are potential risk both to the recipient, their social and sexual context, and to the community. And therefore, we have to have mechanisms, biosecure facilities, if you will, mechanisms to breed animals that are free of known porcine zoonotic organisms, as well as methods to test for the unknown, for organisms that we may not find or may not know about in normal animals. Now, we know, because of testing, that there are not a lot of organisms transmitted from pigs to, say, pig farmers or other people handling pig product, including those of us who've been involved in xenotransplantation. So we think the risk is modest, but we need to be very careful. When we develop these herds, they are called, a term that we developed a long time ago, called designated pathogen free, which is we have a list of organisms and we exclude those organisms from the breeding colony. To do this properly requires, as it does in allotransplantation, a series of assays, both serology or antibody-based tests and molecular, highly sensitive, specific molecular tests as well. We also need to make decisions regarding the porcine endogenous retrovirus, because with the availability of CRISPR-Cas9 for genetic editing, we have the capacity to knock out the more than 50 copies in the genome of each swine of the porcine endogenous retrovirus. So we actually can have PERV-free swine as well, in addition to the various genetic modifications that Raphael and others have mentioned previously. We have specific concerns around certain viruses, such as the porcine cytomegalovirus or porcine roseovirus, because of the fact that these can provoke graft rejection and graft injury, even though these viruses don't spread to human cells. And so we need to think about all of these in the concept of safety in xenotransplantation. That safety requires infection control and occupational health strategies. So as we do these clinical trials and as we follow our most recent kidney transplant at Massachusetts General Hospital, who I just saw 2 days ago and looks great, as we follow these individuals, we need to know what we need to do to protect the staff and that individual and his family. And so what do? Well, we have to develop screening systems that look for both human organisms, because there's a human recipient, and porcine organisms based on the research that we've done. As well, we need to use metagenomic testing to look for unknowns, to think about the idea that perhaps there's DNA or RNA circulating in the individual for which we don't yet know what that organism might be. And we compare those sequences from metagenomic testing to a library of sequences to see if it looks like any other organisms. So far, our testing has been negative. And so we are very confident that we can do a good job in excluding both human and pig organisms in xenotransplantation. Moving forward, we have a lot of very good microbiologic assays to use in screening source animals, to use in screening the xenograft recipient, and to screen, if there's ever any concern, to screen hospital staff and healthy contacts of the recipient, their family, and friends. And so I will stop there with the general idea that we think that we can ensure the microbiologic safety of xenotransplantation so that moratoriums are no longer necessary and that this research can advance with the clinical trials that we're all involved in going forward. So thank you all very much for joining me today, and I'll stop there and be happy to answer any questions.
Thank you, Jay. One question that arises is that based on our research so far, are there any specific organisms that you are concerned about, and they are concerned to the for the recipient and to the general public?
There are no organisms that I'm concerned about for the general public or for public health. That was the original concern about PERV, and it turned out not to be a major concern. The organism that is difficult to exclude from breeding colonies is this porcine cytomegalovirus. And part of the reason is because we need good serologic or antibody tests to make sure that there isn't a distant exposure. It doesn't cause disease in the pigs, and so that they're actually healthy and their molecular testing may be negative. And so we need serologic assays. And what we're able to do is move forward with good serologic testing in the normal animals and then molecular testing in the recipients. But thus far, our testing, and we've done a lot of testing in our recipients, have been unrevealing.
So, I mean, you mentioned designated pathogen-free, and we are hearing about a lot of these facilities being built. Do you think we can exclude a lot of these pathogens by methods that, like what eGenesis have used by knocking out all the proviruses? Is that the reason for the designated pathogen-free?
So the designated pathogen-free is to make sure that the pigs don't acquire new pathogens after being bred to be free of a list of other organisms. So you can decide to knock out all the PERV copies very successfully using CRISPR-Cas9. The introduction of these animals then into biosecure facilities where they're not in contacts with other animals, with rodents or with amphibians or birds, is to make sure that new organisms don't get into the mix, even though they're tested regularly to make sure there's no other organisms. And then they are called, when they are free of that list of organisms, not only designated pathogen free, but we assure that they don't get human organisms and other organisms that might be of concern in the recipient. So the biosecure facility prevents reinfection of these animals rather than after they've already been tested extensively.
Thank you, Jay. We'll have more questions for you in the panel discussion. There are some questions in the Q&A section. We'll address those in the panel discussion also. So moving on to our next presenter, Dr. Robin Avery, who's the professor of medicine in Division of Infectious Disease and Education Director of the Transplant Oncology ID program at Johns Hopkins. And her interest is in hypogammaglobulinemia. And she will talk about the role of IVIG, which has played a significant role in one of the first heart transplant patients. Thank you.
Thank you so much, Dr. Mohyuddin. I want to thank the panelists, organizers, and all of you for allowing me to join you today as a relative newcomer. It's really an honor to present alongside these pioneers in this area. So a couple of questions. Is post-transplant hypogammaglobulinemia a problem? And is IVIG replacement after xenotransplant a problem? If so, what can we do about it? So Dr. Mohyuddin and colleagues reported on the first human xeno heart recipient. As I understand it, on day 47, they had a sharp upturn of anti-pig antibody titers after having received IVIG on day 43. At that time, the recipient had a very low IgG level of 185 milligrams per deciliter. reported to have been lethargic with pulmonary infiltrates and hypotension. And then later, when Dr. Mohyuddin and colleagues went back to examine possible reasons for the rapid onset of diastolic dysfunction and xenograft loss, one of the possible contributors that they looked at was IVIG and the anti-pig antibodies therein. Moreover, they did find binding of several IVIG lots to donor porcine aortic endothelial cells. So why is this a problem? It's led some in the field, as I understand it, to be concerned about replacing immunoglobulin in the xeno recipient at all. But if you look at the left-hand side of this slide, my then colleagues at Cleveland Clinic and I, 20 or 25 years ago, were very, very concerned about the risk of post-transplant hypogammaglobulinemia as an under-recognized risk factor, not only for infections in general, but for severe recurrent constellations of infections, sometimes 2 or 3 opportunistic infections at once. Pictured here, Dr. Bill Braun, transplant nephrology, Steve Mawarder, ID, and Mohammed Yamani, cardiology, are among those who actively investigated this. We found that having an IgG level of less than 400 milligrams per deciliter was a major increased risk for these infections. Then when I came to Johns Hopkins in 2012 and did ID consults in Dr. Bob Montgomery's large program of HLA-incompatible living donor kidney recipients, although there's a survival benefit to that type of transplant, it turns out there's a subset of those patients that also develop severe recurrent constellations of infections and opportunistic infections, aspergillus endophthalmitis, nocardia, severe chronic norovirus, et cetera, et cetera. And together with Jennifer Motter, Dori Segev, Alan Massey, and colleagues now all at NYU, we examined the risk factors for this, and we found that the intensity of desensitization was strongly associated with virtually all of the infections we looked at in total number of infections, which also is associated with prolonged hospitalization and death-censored graft loss. And many of these patients were hypogammaglobulinemic. On the far right-hand side, you see our Johns Hopkins case series of severe chronic norovirus, and we identified hypogammaglobulinemia again as a major determinant of length of symptoms in those cases. So our xenotransplant recipients are going to be receiving, as you know, multimodal immunosuppression. If on top of that, they are very hypogammaglobulinemic, we're quite concerned that they could be at risk for severe, again, recurrent and opportunistic infections. Using a term coined by my late mentor and Jay's, Dr. Bob Rubin, their net state of immunosuppression is going to be of nearly unprecedented magnitude. Is there some way we can mitigate this risk? So we have 2 thoughts about how this might happen. On the right-hand side, you see Dr. Maria Bettinati, our Director of Immunogenetics at Hopkins, and Dr. Kaz Yamada, who leads our xenoimmunobiology research group. They and colleagues are working on devising better ways of screening lots of IVIG to try to select those with the lowest anti-pig levels and reserve them for later use. However, in our recent letter to Transplantation, my colleagues and I, pictured here, Olivia Cates, Kapil Saharia from University of Maryland, Brian Lindner, John Badley, Andrew Caraba, we're actually calling for a more radical potential solution, which is the intentional development of an IVIG product that eliminates anti-pig antibodies as much as possible. There is work from some years back, as you probably know, that actually suggests this is possible. And even though it would take years to develop and test and gain regulatory agency approval of such a product, we propose that this would actually contribute significantly to the xenotransplant enterprise as a whole and to the safety of our patients, protecting them from these severe infections. And I'd like to thank my colleagues, Dr. Andrew Cameron, Director of Surgery at Hopkins, who's leading our 10GE Xeno Kidney clinical trial in conjunction with NYU and United Therapeutics, and Dr. Kazuhiko Yamada and lab, whose work in non-human primates is the underpinning of this clinical trial. Thanks to all of you as well who are working in this area and making it safer for our patients. Thank you.
Thank you, Robin. So one of the question is that is the IVIG replacement associated with the risk of xenograft rejection? Do you think we can find an alternate to that?
So Mohammed, I'd be really interested in your take on that, since that beautiful paper that you and your colleagues published in The Lancet examined that in detail. You found, I think, binding to endothelial cells, but you did not find associated complement-dependent cytotoxicity. Is that correct? Would you like to comment on that maybe?
Yeah, I mean, so there there was a lot of binding, both IgG, IgM, in different lots, but it varied from lot to lot. And yes, we did not find a, have to revisit to see if we found any CDC from that. But I think there was some amount of CDC, but not, based on the amount of antibody we saw. So, I mean, this is something that we need to, explore further. Let's move on to our last presenter, Dr. Leo Buehler. Leo Buehler is the editor-in-chief of Xenotransplantation. He's also the past president of the International Xenotransplant Association. He's the chair of our organizing committee of our upcoming International Xenotransplantation Congress in Geneva in September, where you all are invited. And he's a professor of surgery, and he'll talk about the regulatory affairs involving xenotransplantation. So, Leo, take it away. You are muted, Leo. We should hear you now.
Can you see the slides?
Yes. Can you
Good.
put them on in presentation mode?
Here we go.
Thank you.
Thank you very much. So thank you for the organizer to invite me. So I will go back to a topic, which is the collaboration with WHO. Wayne has already mentioned that IXA has a long history of collaboration with WHO, and I will go back to the initiation and the start of this collaboration. So the WHO has each year, as you know, the General Assembly that takes place here in Geneva. I'm located in Geneva. And all the ministries of health come together for a couple of weeks and discuss and approve important topics. And the first time that xenotransplantation was mentioned in a WHO document was in 2004. And during the General Assembly of 2004, an update of transplantation also wanted to include xenotransplantation. And you see here the guideline document that's what approved and then sent to all the ministries of health around the world. And interestingly, xenotransplantation was supported by WHO, especially also to solve the shortage of organs and organ trafficking. So WHO was supporting from start xenotransplantation as a solution to the shortage of organ, but realizing the risk of xenozoonosis was, of course, advocating for a regulation and a supervision of all the regulatory agencies around the world. So in 2004, IXA contacted WHO. At the time, it was Dr. Luc Noël from France, who was in charge of the transplantation office at WHO. And we organized in 2005 a first meeting with a WHO representative and about 20 colleagues from all the regions around the world to discuss what was going on in xenotransplantation. And it was realized that xeno activities were ongoing in several countries, and most of the time with no regulation. So the idea was to start inventory. We chose inventory, not to use the word of registry, because the data that we collected were very limited. And we started in collaboration with WHO and IXA, inventory based in Geneva that was collecting data to document what was going on. And we started about 10 years before 2004, including all the recent activities that had happened. For example, the cow growth activities with pig islet transplants to over 10 patients in Sweden, etc. So we collected all these data. and put them with limited knowledge, with source animal, regulation, etc., in this inventory. And the inventory was put online in 2005, so quite exactly 20 years ago. And on a regular basis, we published in Transplantation an update on these activities. And you can see here, for example, just here, the activities. You see here the the source animals, mostly pigs. But at the time, there were sometimes sheep or even fish for skin, etc., used around the world, often without regulation. So, for example, here you have the information if this activity was under a national regulation. And you can see here that before 2004, there was often no regulation in the countries where this happened. And just around after this opening of the inventory, you can see here that since 2005, all the activities that we could include in this inventory were approved by a national regulatory agency. So this is very important. And you can see here that since 2000 to around 2000, so over 20 years now, activities have been ongoing, initially mainly skin or tissue, islets, and only about since 2021, as Raphael just presented, heart and kidneys have been performed. So what is the future? So we have ongoing contacts with the WHO. As Wayne mentioned, there have been several meetings after this 2005 meeting in Changsha, in China, have been organized WHO consultations. And we hope that in September, just before our next international congress in Geneva, we will be able to co-organize with WHO a meeting to update several topics. And one topic will be this inventory, because this inventory is still quite limited in data. And one question we have for WHO, if this inventory could be included in WHO ongoing registry for organ transplant, which is the global observatory that is held in Spain, but for WHO. And one possibility that we have started to discuss is if our inventory with quite limited data could be in the future, and also now that the activities increase in numbers, if this collection of data could be included in a larger inventory or registry like the Global Observatory of WHO. And this hopefully will also be discussed during our next Congress. And I invite all the interested people in xenotransplantation to join, if they can, the next Congress in Geneva, September 30th to October 3rd. Thank you.
Thank you, Leo. You mentioned about the WHO oversight. Do they have any opinions so far, or they are still working on it?
With the contacts we had on a regular basis with WHO, and we had also meetings in China with WHO delegates. WHO is continuously supporting xenotransplantation as a solution for organ shortage. However, due to the risk of infectious diseases, regulatory agencies must oversee the activities with regulatory restrictions that guide the choice of animals, the choice of the recipient, and then the long-term surveillance of patients and their close contacts. But WHO is in favor of activity of xenotransplantation, of initiation as a solution to organ shortage.
And how is IXA helping, get this oversight, right? I mean, you don't want xenotransplantation to go out of control and without any oversight. What is IXA doing in that regard?
I mean, we have different groups, subgroups, and we put out white papers. So our proposal, our guidelines are included in these papers that just came out in transplantation and xenotransplantation.
Yeah. So now we can open this, the forum for everyone, and we'll take questions from the Q&A section. We have some questions sent to us before. Wayne, I would ask that question again from you because you've been spearheading the Changsha communication. So what's the objective behind that?
Yeah, so I was going to bring that up to follow up from Leo. The IXA, as Leo oversighted in conjunction with TTS and WHO, developed the Changsha Communique. The Changsha Communique was basically a guidance document that was developed in response to international bodies, including the regulators, and the development of the Changsha Communique also involved regulators from all countries around the world to ensure that we had a consistent standard and set of guidelines for all regulators in the world to follow so that we could have a uniform approach to how we undertake clinical xenotransplantation. And WHO felt that IXA, as the expert body, were best served to give those opinions, and thus supported the formation of the Changsha Communique. And that's why it's been important to update it on a regular basis, and that's why we've been pushing since 2018 with the very recent surge in xenotransplantation and types of pigs, as well as immunosuppression, as well as screening activities, to update the Changsha Communique, or as we should call it, the IXA guidance document to regulators to ensure that we continue to update those guidelines with expert opinion.
Yeah, so I would like to ask Jonathan to, address a few questions that have been put up in the chat box, in the question and answer.
Okay. I can't see the other questions, but there's one question still remaining. How is the pig donor selected based on the recipient? Are they bred for different blood types? Besides microbiological testing to prevent infectious disease transfer, is there a standard battery of histocompatibility tests done before the organ is recovered from the pig? So a couple of our panel could answer that, please.
I guess a good starting point would be basically there's been a number of various groups around the world that have been looking at different organs for transplantation. So I think picking organs that have been transplanted, certainly the hearts and kidneys, versus what we do, islet cell transplantation, there can be differences in the transgenes. The uniform thing that everyone's discussed tonight or today is that the alpha gal epitope or any of the gal is knocked out. A number of other transgenes can be expressed to try and prevent coagulation, complement upregulation, and now with immunosuppression, such as in islet cell transplantation, you can genetically engineer towards those organs. And certainly you may need less, and we feel certainly for islet xenotransplantation, you don't need all 59 transgenes. Or variants of genetic alteration, but you may need a key set of at least 3 to 5 for islets. The kidneys, and I think Raphael can probably answer the relation to kidneys, and others can relate to other organs, but certainly we try and engineer the organs also for size, certainly for things like heart and liver. And so there's a particular transgene that moderates the size, because pigs can grow to extremely large sizes. So we have to moderate their growth as well. So Raphael, do you want to buy in for
Yes.
that?
I think part of the question was with the ABO groups, correct?
Yeah.
And this is probably, I'm speaking under the supervision of Dr. Mohideen, but I think that the pigs, they have an AO's blood group system, but basically they are engineered in one way so they can be used in all the patients. There is not
Yeah,
yet a
both group are.
personalized genetic, and I think they all behave as O at the end,
That's
because
right.
we remove alpha gals and all the other things.
Yeah.
So no, there is no like specific matching so far. And I think part of the question was also if we perform specific testing for like cross match and such. And yes, we do. We do cross match. We measure anti-pig antibodies, and this is one of the most important, because we're not all equal facing that, because some patients are hyper already immunized and sensitized against pig antigens. I don't know. And regarding size, yes, we have two ways of dealing with that. Is one if the growth hormone knockout, and the other one is to breed the pig in a miniature pig background, like the MGH team developed. Mohammed, do you have anything to
Yeah,
add to
no,
that?
so no, I mean, in the interest of time, we should move on. There were several questions that you and others have answered in the question answer session, but not the entire. It's not open to the entire audience. So I would repeat some of those questions. So one question was that it may be an odd question: does the sex of the pig matter in donor selection? Whoever wants
I
to take
think that was directed to me. So I would say that's an excellent question. That's not an odd question. I do believe that we mostly used males for transplant and experimentation, because they have less variations with hormones and such. But I think that's going to be an interesting question to answer in the future. We do not know yet if there is a strong impact or not, I would say, to be careful.
So from our heart experience, I would say that, we used the pig of the same size in both patients, but the female heart was much smaller than the male heart. So I think that is a great question. Jonathan, you have other questions?
Can I buy in on that one as well,
Sure,
Mohammed?
sure.
Certainly, from a islet transplant perspective, it doesn't matter what sex, and we use multiple litters of piglets, so we use male and female. So I guess that would be more targeted to solid organ.
Well, on our 3 minutes left, I think there's one question that's been answered, but some more discussion would be useful. The question was, what strategies have been employed to overcome acute and chronic rejection, as well as physiologic barriers in pig-to-human xenotransplant? I'm sure you could spend an hour and a half on this, but if you could sum it up in 2 to 3 minutes.
I mean, this is a great question for anybody here. I mean, we removed the alpha gal. That was the first process, and then there are different antigens that were knocked out that are causing hyperacute rejection due to perform antibodies, and this is what we call triple knockout, because other enzymes that produce those antigens were progressively knocked out. And then I think part of the transgenes helps whatever is related to complement regulation is helpful to prevent that. And then specific immunosuppressing regimen were developed. Dr. Buehler, who is here, helped to develop in 2001 and emphasized the importance of co-stimulation blockade, which is very important, we believe, in xenotransplantation. So those are a couple of answers to that question. I'm sure others can add more.
I might add to that despite the use of co-stimulatory blockade and all the modifications that Raphael just referred to, we've had some rejection in our early xenotransplant recipients. And so the final package of immune suppression, we don't know yet. Our current recipient's doing quite well, as I mentioned earlier. But I think that we need to be refining, and we may need techniques to better assess the immunity in each individual going forward. And so metagenomics can help us with that. Other assays can help us with that. But we don't have the final package yet. But these individuals are heavily immunosuppressed for T cell, B cell complement, in addition to the 10 gene modifications and the perf knockouts. So I think it's important to know that we haven't quite gotten there yet.
You've seen some ACR in both your patients, if I'm not mistaken, and that is fascinating to, I think, most of us, because we focused on antibody-mediated aspect. But how can you expand briefly on that? Why did we see this ACR that was actually reversed in both patients very early on?
Actually pretty easily reversed with standard immunosuppression, steroids and some T cell depletion. But these individuals had been T cell depleted going forward. I think part of the problem is that with the immunosuppression that we use now, which is calcineurin based, we may deplete regulatory T cells, and we may not have the best combination. And there are some new agents that preserve regulatory T cells while doing T cell depletion. So as I say, I don't think we're quite there yet, but as you say, we've seen early rejection in those, just as was seen in some of the early clinical trials of close stimulatory blockades. So we're not quite at the finish line yet.
So Jay, I mean, there was one question. Have you seen any BK virus or CMV in any xenokidney patients?
Yeah, so very good question. I think we're in the process of publishing our data, but our first patient had prior kidney transplant and actually had both CMV and BK and EBV infections in his previous allograft, with his previous allograft. These were easily treated and actually detected by metagenomics, but did have some early reactivation when we treated his graft rejection. So we did see a little bit of BK and EBV in that individual. And therefore, we have to screen for both human organisms that are common as well as pig organisms. They didn't cause any trouble in our recipients, but it's certainly important to be aware of that potential.
So what I'm hearing from this panel is that there's a need for further gene modification. There's a need of modification of our immunosuppression because our conventional immunosuppression doesn't take care of everything we are seeing in our xeno patients, right?
I think that's fair, whether it's approached through genetic modification or through exogenous immune suppression. It hasn't yet been determined. I think, and the concern is always that the more we immunosuppress these people, as Robin referred to, is that we increase the net state of immune suppression and their infectious risks go up, even from infections acquired in the community.
So, I mean, a question that's asked very commonly is like, what is success in xenotransplantation? How do you determine success? Anyone?
I guess it would parallel that of the early days of allotransplantation. The early days when the first kidney transplants were performed and were, as we then said, successful. Some of them are only very, very short term. We're dealing with very, very sick patients to start with, a cohort of patients that won't necessarily get allotransplants. Like the allotransplants that were done in the early days, we will see issues with the cohort of patients that we'll move forward with. So I think success is, like we've already seen, it's going to be a challenge, but any step forward, certainly to get patients to, even like in your patient, 60 days was a major milestone. That's fantastic outcome for the whole field of xenotransplantation, and a point at which we start to then move forward. And certainly looking forward with the kidney transplants now, we would call that a great success. So we can build on it the same as what we did with allotransplantation in the past. We'll see things improve and hopefully get longer graft survival, and more importantly, have patients that live and end up with long-term a graft for life potentially.
I think one final question from the chat is that you know how IXSAFE is formally engaging the global community, patient community, so that they are active stakeholder in the development of xenotransplantation.
Maybe I'll start with that one because it builds on what Wayne just mentioned, which is that our patients are the people we serve, and their expectations, getting them past organ failure with a xenograft, is really what we aim to serve. What we want these people to be able to do is get back to the life that they desire. And so the IXA is centrally involved in making this safe and in sharing information so that everyone is starting from the same place. And as Wayne referred to with the guidelines, at least we can assure the basic safety and communication of the available information so that the whole field can move forward. But the only people who really matter in this whole equation are the patients, and therefore we are starting to engage patients in surveys and in other areas that have been published, both in transplantation and in xenotransplantation, which are attitudes both by professionals and by the public in terms of xenotransplantation. We've got to keep our eye carefully focused on that to make sure that we're serving the right people. It's our patients. Although the research is fascinating, it really is ultimately about our patients.
I think you covered it very well, and we are already over time. It was a great discussion. I would like to thank all the panelists, my co-chair, Dr. Bromberg, and also Jennifer Lee and Jennifer Government for putting this together, which was a very timely webinar. I hope you all have learned a lot from this webinar. The discussion should continue, and we again would like to invite you to come to International Xenotransplant Association Annual Congress in Geneva, and hope to see you all there for further discussion. Thank you very much.